So, good day, Doc. Now I will be discussing on prion diseases and human cancer viruses. So, what are prion diseases? Prion diseases are also known as transmissible spongiform encephalopathies, or TSEs, which are a family of rare progressive neurodegenerative brain disorders that affect both humans and animals by eating food contaminated with them. Prions are abnormal forms of normal proteins because they're single molecules containing approximately 253 amino acids. Proteins are long chains of amino acids that fold together into a unique shape to help a cell function in a particular way. The normal and harmless prion protein is found throughout the body but is most abundant in the nervous system. Prions have the ability to convert the normal forms that they come into contact with into abnormal forms. No antibiotics can cure disease caused by prions. And the reason why prions are so hard to treat is because, unlike bacteria and viruses, prion diseases are irreversible and cannot be overcome by the body's immune system. Prion agents are very resistant to inactivation, including formaldehyde and boiling and radiation, although they can be inactivated by bleach and autoclaving. So prions are not destroyed even if the beef or material containing them is cooked or heat treated. The normal harmless prion protein is usually designated as PRPC. C stands for cellular, and the abnormal infectious form, which is the disease, is PRPSC. SC stands for prototypical prion disease scrapie. So, diseases that are caused by these abnormal proteins are as follows: scrapie, bovine spongiform encephalopathy, or BSC, and chronic waste diseases in animals, while in humans, the Kuru and Creutzfeldt-Jakob disease. Scrapie is a fatal degenerative disease affecting the nervous systems of sheep and goats. It is one of several transmissible spongiform encephalopathies, and as such, it is thought to be caused by a prion. Chronological disease, meaning it usually lasts for a long time. The causative agent is due to the misfolding of protein or prion proteins. In early infections, infectivity can be recovered from lymphoid tissues. High titers of the agent are found in the brain, spinal cord, and eye, the only places where pathologic changes are observed. A titer is a laboratory test that measures the presence and amount of antibodies in blood. A high titer could show residual antibodies to a previous infection or unsuccessful attempts to bond to the antigens in the bloodstream by the antibodies. So the bovine spongiform encephalopathy, or BSE, is commonly known as mad cow disease. It is a neurological disorder commonly found in adult cattle. First recognized in Great Britain in 1986, it is transmissible. It is a transmissible disease that affects the central nervous system, and it may be passed to humans who have eaten infected flesh. This disease is just like any other diseases that are caused by prion proteins, like the Creutzfeldt-Jakob disease in humans, scrapie in sheep, and chronic waste disease. The spread is caused by feeding rendered material from infected cattle back to another cattle. We all know that cows are herbivores, but they can also eat meat, so this makes them susceptible to this disease. The BSC disease has a long incubation period, which is about 2 to 8 years. Clinical signs of this disease are found in adult animals. Symptoms may last for a period of 2 to 6 months before animal dies. Animals with mad cow disease may demonstrate some of the following symptoms, which are the nervous or aggressive behavior, depression, hypersensitive to sound and touch, abnormal posture, weight loss, difficulty rising from a lying position, decreased milk production. For prevention, there is no effective vaccine yet, although taking measures like banning the meat where the disease is present is possible. Scrapie-like disease that is found in mule deer and elk in the United States and Canada. They are laterally or directly transmitted with high efficiency from animal to animal via saliva, urine, feces, artificial feeds with mammalian proteins. Indirect transmission can happen from soil to animal. There is no evidence of the disease being transmitted to humans or livestock. Signs and symptoms include emaciation or thinness, excessive salivation, lack of muscle coordination, difficulty in swallowing, excessive thirst, and excessive urination. So, as you can see, this is a picture of the areas of infection. So, the brain, eye, spinal cord, tonsils, spleen, and lymph nodes that are also around the digestive tract are the areas that are infected by this disease. Two human spongiform encephalopathies include the CRU and the classic form of Creutzfeldt-Jakob disease, or CJD. CRU disease only occurs in the eastern highlands of New Guinea and was spread by customs surrounding ritual cannibalism of dead relatives. So in that place, during the 1950 to 60s, family cooked and ate dead family members, body tissue, and brain. The Creutzfeldt-Jakob disease can be transmitted from an infected person to others, but only through an injection or consuming infected brain or nervous tissue. CJD in humans develop gradually with progressive dementia or Alzheimer's disease, ataxia, where people lose muscle control in their arms or legs, and myoclonus, which is the sudden twitching of muscles, and eventually leads to death in 5 to 12 months. So the word crew is derived from a term in the Fore language that means to shiver or tremble in fear. The disease led to the creation of a new class of diseases, including Creutzfeldt-Jakob disease, Gertzmann Straussler-Scheinker disease, and fatal familial insomnia. Symptoms included difficulty walking, poor coordination, difficulty swallowing, slurred speech, moodiness, and behavioral changes, dementia, muscle twitching and tremors, inability to grasp objects, random compulsive laughing or crying. Creutzfeldt-Jakob disease, or CJD, is a rare, rapidly worsening brain disorder that causes unique changes in brain tissue and affects muscle coordination, thinking, and memory. There are about 350 cases per year in the US. The two main symptoms of this disease are several mental deterioration and dementia, and involuntary or unwanted muscle jerks or muscle movement. Early symptoms of the disease may include lack of coordination problems with walking and balance, behavioral changes, depression, mood swings, and anxiety, and insomnia. As CJD progresses, symptoms may include weakness of the arms and legs, blindness and loss of the ability to move or speak, pneumonia and other infections, and coma. Some symptoms of CJD can be similar to symptoms of other progressive continuing to worsen neurological disorders such as Alzheimer's and Huntington's disease. However, these symptoms tend to worsen faster in CJD than in Alzheimer's disease or most other types of dementia. So, what are human cancer viruses? These are viruses that are capable of causing cancer in humans. Most are DNA viruses except some retroviruses and flaviviruses, which are RNA viruses. They influence the cell cycle by encoding proteins that direct cell cycle progression and integrating near cellular genes that control cell cycle progression. So here we have the viruses associated with human cancers. These are the viruses that cause cancer in humans. They are the papilloma virus, herpes virus, hepatitis B virus, polyomavirus, retrovirus, and flavivirus. Here we have the tenets or principle of viral carcinogenesis. Viral carcinogenesis refers to the process of transformation of normal cells into cancer cells induced by a virus. So, viruses can cause cancer in animals and humans. Tumor viruses frequently establish persistent infection in natural hosts. Host factors are important determinants of virus-induced tumorigenesis. Viruses are seldom complete carcinogens. Virus infections are more common than virus-related tumor formation. Long latent periods usually elapse between initial virus infection and tumor appearance. Viral stains may differ in oncogenic potential. Viruses may be either direct or indirect acting carcinogenic agents. Oncogenic viruses modulate growth, control pathways in cells. Animal models may reveal mechanisms of viral carcinogenesis. Viral markers are usually present in tumor cells. And lastly, one virus may be associated with more than one type of tumor. So oncogene is the general term given to genes that are involved in cancer causation. The cellular oncogenes are abnormally expressed or mutated forms of the corresponding proto-oncogenes. As a consequence of such alterations, the oncogenes induce abnormal cell proliferation and tumor development. Cellular oncogenes are partly responsible for the molecular basis of human cancer. While suppressor genes, a second class of human cancer genes, is involved in tumor development that are negative regulators of cell growth. They form complexes with oncoproteins of certain DNA tumor viruses. Examples are the retinoblastoma and p53 gene, which blocks cell cycle progression. Tumor viruses establish long-term persistent infections in humans because of differences in individual genetic susceptibilities and host immune responses. Viruses that establish persistent infections must avoid detection and recognition by the immune system that would eliminate the infection. It is believed that host immune surveillance mechanisms usually eliminate the rare neoplastic cells that may arise in normal individuals infected with cancer viruses. However, if the host is immunosuppressed, cancer cells are more likely to proliferate and escape host immune control. Neoplastic cells are abnormal growth of cells or tissues in the body leading to cancer. So here we have the papillomavirus. Papillomaviruses are circular double-stranded DNA viruses of 8 kilobase. They are icosahedra in shape, 55 nanometer in diameter. They have two structural proteins, L1 and L2. Here, L1 and L2. Human papillomavirus, or HPV, is the most common STI. Transmission of viral infections occur by close contact. Papillomaviruses cause infections at cutaneous and mucosal sites, sometimes leading to the development of different kinds of warts, including skin warts, plantar warts, flat warts, anogenital warts, pharyngeal wart, papillomas, and several cancers, including those of the cervix, vulva, penis, and anus, and a subset of head and neck cancers. Estimated 660 million people worldwide have HPV genital infections. HPV occurs in adolescents and young adults younger than 25 years. They are known as the cause of anogenital cancers, which include anal, cervical, penile, vaginal, and vulvar cancers. Widespread use of the Papanicolo or baby smear for cervical cancer detection has led to decrease in deaths due to cervical cancer. The cytological test aims to detect precancerous cellular changes in morphology, allowing for lesion removal prior to cancer development. Vaccines against HPV to prevent cancer-causing infections, and the use of protection during sexual contact, because that can protect women from HPV infection. For treatment, there is no treatment for the virus itself. However, precancerous lesions need to be removed through surgery. So here we have the retroviruses. They are diploid single-stranded RNA viruses, 7 to 11 kilobase. They're spherical and 80 to 110 nanometer in diameter. They're helical nucleoprotein and enveloped. They are divided into 7 genera: the alpha retrovirus, beta retrovirus, gamma retrovirus, delta retrovirus, epsilon retrovirus, pumavirus, and lentivirus. They can be exogenous or endogenous. Exogenous retroviruses are spread horizontally and behave as typical infectious agents after contact. So basically, they are pathogenic retroviruses. Horizontal disease transmission implies that pathogens are spread to other members of the same or different species through non-hereditary means, while endogenous retroviruses are transmitted vertically, meaning they're spread from parent to offspring. They affect cellular gene expression patterns. Important features of endogenous viruses are as follows. DNA copies of RNA tumor virus genomes are covalently linked to cellular DNA and are present in all somatic germ cells in the host. And then the endogenous virus may be induced to replicate either spontaneously or by treatment with extrinsic or chemical factors. So, major determinant of the host range of a retrovirus is the presence or absence of an appropriate cell surface. Infection occurs by the interaction between the glycoprotein envelope and a cell receptor. So, a major determinant of a retrovirus's host range is the existence or absence of an appropriate cell surface receptor. A cell surface receptor and the viral envelope glycoprotein interact to start an infection. Only animal cells from the original host species are infected by and used for replication by ecotropic viruses. Amphitropic viruses have a wide host range because they can infect cells from both their normal host and heterologous species because they recognize a widely used receptor. And then in some heterologous or foreign cells, xenotropic viruses can multiply but not in cells that have been cultured from the native host. So the host ranges of numerous endogenous viruses are xenotropic. So the retrovirus life cycle could be divided into 7 steps. The first is binding, and then fusion, reverse transcription, integration, replication, assembly, and budding. So for prevention, screening blood products for antibodies can be done, and education to prevent transmission by sexual contact or the sharing of needles. Next, we have the hepatitis B virus of the Hepadnaviridae family. They are 42 in nanometer and spherical envelope virions. They are circular and double-stranded DNA, approximately 3.2 kilobase. Hepatitis B is a risk factor in the development of liver cancer in humans. Epidemiologic and laboratory studies have proved persistent infection with hepatitis B virus to be an important cause of chronic liver disease and the development of hepatocellular carcinoma. Infections occurring in adults are usually resolved, but primary infections in neonates or neonates means newborns and young children tend to become chronic in up to 90% of cases. This virus is usually spread through bodily fluids. However, children become infected with this virus if they are born to a mother with the virus. Older children can become infected through injection, drug use, or unprotected sex. The hepatitis B vaccine that made prevention of primary infection raised the possibility of prevention of hepatocellular carcinoma, particularly in areas of the world where infection is hyperendemic, like, for example, Africa, China, and Southeast Asia. So next we have the herpes virus. Herpes virus are linear double-stranded DNA viruses, 125 to 240 kilobase. They're icosahedral in shape, 125 to 200 nanometer in diameter. They are envelope virions. Herpes viruses have been linked to several specific types of tumors. They cause acute infectious mononucleosis when it infects B lymphocytes. EBV can cause Burkitt lymphoma or BL. It is a tumor most commonly found in children in Central Africa. To nasopharyngeal cancer, which is common in Cantonese Chinese and Alaskan Eskimos. And to post-transplant lymphoproliferative disorders and the Hodgkin's disease. Lastly, the polyomavirus is circular double-stranded DNA virus of 5 kilobase. They are icosahedral and 45 nanometer in diameter. They're non-enveloped and replicate in the nucleus. So the two well-known human polyomaviruses are BK virus and JC virus. These viruses were first isolated in 1971. JC from the brain of a patient with progressive multifocal leukoencephalopathy or PML, and BK from the urine of an immunosuppressed renal transplant patient. This infection usually occurs during early childhood, and both viruses may persist in the kidneys and lymphoid tissues of healthy individuals after primary infection. BK viruses cause hemorrhagic cytosis in bone marrow transplant recipients, while JC viruses is the cause of progressive multifocal leukoencephalopathy. Um, hemorrhagic cystitis is a condition in which the lining of the bladder becomes inflamed and starts to bleed. So the blood can be seen in the urine, while progressive multifocal leukoencephalopathy is a disease of the white matter of the brain caused by a virus infection or the polyomavirus that targets cells that make myelin. Myelin is the material that insulates the nerve cells or neurons. So that'll be all for my reporting. Thank you.